Welcome back to Psychedelics Today, everybody. This is Joe Moore coming at you from Breckenridge, Colorado. Today on the show, we have Dr. Brian Pace. It's the first time I've had to say that to the microphone. Dr. Brian Pace. Brian just became doctor, a few weeks ago. And this interview was recorded maybe a month or two back with Dr. Jason Slott. So this is actually a special episode. Brian actually steps in as a special guest and interviews Dr. Jason Slott about all sorts of interesting stuff. They talk about psilocybin developing in mushrooms. Maybe I could start with this. Jason Slott, assistant professor of fungal evolutionary genomics. This could be an old thing, but from Ohio State University. Looks like he finished his postdoc work at Vanderbilt. And yeah, Brian, having a strong background in evolutionary biology, talking to Jason Slott, who also does. It's a really great conversation. I'm not an evolutionary biologist, so I can't really say too much here, but they go all over the map about psilocybin, fungi, evolutionary dynamics there. And I think it's a story worth knowing about. So let us know what you think. Hit us up over at Psychedelics Today, email at gmail.com. And yeah, expect more. Hopefully we can get Brian to help us out a little bit. I think he's working on one or two more interviews for us. That'll be really cool once we can secure that. And I can't wait to get out to Columbus, actually. They're all in Columbus at OSU, and I actually just got a wedding invite. So looks like I'm going to be in Columbus at least once in the next year. So excited to check out the Ohio scene. If you want to support the show, just let a friend know. Shoot us or send them your favorite episode or leave us a review at Facebook or iTunes. We'd love it. Hope you enjoy this episode of the show, and see you on the other side.
Hi there. My name's Brian Pace, and I am here with Dr. Jason Slott at the Ohio State University. Today we're going to be talking a little bit about Dr. Slott's contributions on the topic of the Psilocybe genus and psilocybin. So Dr. Jason Slott is faculty at OSU in the Department of Plant Pathology. Jason, can you tell us a little bit about how you came to Ohio State?
Sure, and thanks for inviting me, Brian. It's really fun to be able to talk about my research. How did I get to Ohio State? Well, there's not a lot of mycology job postings around. I was searching around for a couple of years during my postdoc, studying mushroom evolution, and the perfect job all of a sudden came up at Ohio State, looking for somebody studying the evolution of fungal genomes, which was pretty target with what I had been researching. So I interviewed here, and they liked my research, and I started working.
Great. So before we get into some of your more specific work on psilocybin and its evolutionary history, I wanted to back up a little bit and talk about some of the ideas that you think about in your lab more generally. So you have a page on your lab website about neurochemical ecology. I was wondering, what exactly can you tell our listeners what neurochemical ecology means to you?
Sure. So ecology, we all know, is the interaction among organisms, the study of the interactions among organisms and their environment. Well, chemical ecology is how certain chemicals, certain molecules are the mediators between these different organisms, us and plants, for instance. Now, neurochemical ecology is a special case of chemical ecology where the chemicals that we're talking about are specifically targeted to processes in nervous systems of animals.
Okay, fascinating. I think that when we think about neurochemical ecology, the idea that there are these molecules that interact with our nervous system, altering our perception. Most of us think of various recreational or medicinal substances, but neurochemical ecology is altering the perception of animals and mostly animals, right?
Well, that's how I've proposed the idea, is that it's plants and fungi producing chemicals that directly target specific parts of the nervous system of animals. But animals is a pretty large category when you think about it. And we're not only talking about humans and other mammals, but we're talking about insects, probably the number one, the most widely distributed and most diverse animals on Earth are insects, nematodes, these sorts of things. So there's a broad opportunity for different plants and fungi and bacteria to exploit worms and insects and potentially even mammals by producing new drugs and other compounds that sort of either manipulate their behavior or just inhibit their feeding, or potentially getting them to disperse their spores, disperse their seeds, for instance.
Interesting. So, I mean, I think that most people, when they think about fungal metabolites, if they ever do, the one that has really stolen the show has been psilocybin. It's something that came into Western consciousness after Maria Sabina's encounter with Gordon Wasson in the 50s. It was shared in Life magazine, and that was most people's first introduction to the idea that there were mushrooms that could alter one's consciousness. So do we have an idea now? We were talking about the diversity of insects, but there's quite a bit of diversity that's fungal. And do we have any idea of how many species might be producing psilocybin? Are they all in the Psilocybe genus?
Well, no. And the answer that I would have given you 6 or 8 months ago is different than the answer that I'm going to give you now. So previously we thought there were about approximately 200 species of mushroom-forming fungi belonging to just a handful of genera, Psilocybe, Paneolis, Gymnopolis, and even a Pluteus, which looks an awful lot different than those mushrooms. About 200 species distributed amongst those genera, and even potentially in one lichen-forming fungus. But in the past 6 months, I've come to find out that there are also very different kinds of fungi that infect insects that are producing psilocybin, in addition to other neuroactive compounds.
So when you say, well, maybe we can get into some of the ones that are interacting with insects a little bit later, because it's a fascinating story. But listeners may be aware that there are definitely complex interactions between fungi and insects. I believe, if I remember from one of my mycology textbooks, there are beetles that will have specialized structures on their exoskeletons for collecting mycelium that their larvae are obligately they must have this mycelium to grow and live. So we can see that there are tight mutualisms with plants and insects, just like many people may know that there are ubiquitous mutualisms between plants and fungi in the form of, say, mycorrhizae. But Psilocybe species they're widely distributed. They're as far as I'm aware from reading Paul Stamets's Psilocybe mushrooms of the world, or psilocybin mushrooms of the world, I think it's called. There are samples of Psilocybe in particular on every continent, including Australia and New Zealand. I imagine that if Antarctica was not frozen, we might find some there. So I was curious if you could talk a little bit about this, since evolutionary history is a topic that I think we both share a fascination in. And I was curious if this wide distribution is because they were distributed before the breakup of supercontinents like Pangea, or that they followed the radiation of animals.
Well, I think that the answer is probably all of the above. There's a very complicated history of how the ability to produce psilocybin has evolved. Mushrooms were pretty well anatomically modern when you had a large supercontinent. There's a fossil of a mushroom that looks an awful lot like psilocybe from about 115 million years ago, at the time when most of the continents were together in a supercontinent called Gondwana. That's the oldest mushroom fossil, but you can imagine if psilocybe, or the ability to make psilocybin, was around then in the forests that were much warmer environment, much more uniformly warm environment. You had flowering plant forests that went almost all the way up to the poles. And there's just a huge amount of production of wood and fungi. Specifically, these groups of fungi that we're talking about are really good at breaking down wood. They pretty much invented all the best ways of digesting wood. And at some point in maybe 130, 140 million years ago, they started coming into competition with wood-eating insects like termites and cockroaches and ants. And the termites really probably gave them a run for their money. And but anyway, I'm sort of getting ahead of myself there. The continent, the supercontinent, would have broken up after that. Certainly after the KT extinction event, the continents really separated a lot, and those mushrooms would have been distributed amongst the continents. But also, there's definitely examples where, and I don't really have any specific data that I can talk to you about that, but there's probably a good argument to be made that humans have been distributing some psilocybe fungi in livestock, as some of these fungi grow in the dung of horses and cattle, which humans would be bringing around the world. And also migrations of these animals have gone over land bridges, for instance. They could connect continents at much more recent time frames. So there's probably not only been a spreading from these mushrooms existing in the supercontinents, but also multiple long-distance dispersals.
Yeah, so I think that's a fascinating topic. I want to perhaps dig around and explore different aspects of it. For instance, more recently, in terms of evolutionary history, I'm aware that when an event called the Columbian Exchange happened, essentially when we had colonization from the old world to the new, there was one of the biggest exchanges of species of plants and fungi and diseases. And among some of those species that came from the old world to the new, from Eurasia to the North and South America, was actually Psilocybe cubensis. And so, while some of us may be familiar with the story of Teonanactl, the flesh of the gods, that these psilocybin-containing mushrooms were used for spiritual and religious purposes among the Aztec and possibly Maya, that they weren't using the cubensis that most people are familiar with. They were using Psilocybe mexicana. And so there's these recent introductions that are human mediated, but you're talking about really vast periods of evolutionary time where psilocybe was moving throughout the world through all the methods that you talked about. So you mentioned that you didn't have much data, and I'm not sure I've come across any myself about how psilocybe or dung rotting mushrooms may have been affected by the rise of animal domestication. But I'm wondering, is this something that we can sort of walk through mentally here and imagine what might happen as a group of primates starts domesticating ruminants that are producing dung in higher concentrations in a geographic area? How might that affect the adaptation and evolution of something that uses dung for food?
Well, it's obviously, if we are increasing the numbers of cattle that we're tending, or if we're following expanding herds of ruminants that are taking over emerging grasslands, then the amount of dung is going to increase. So if the amount of dung increases, there's an opportunity that opens up, and I like to think of evolution somewhat in terms of filling niche spaces, taking advantage of opportunities. And so if the biomass of herbivorous mammals is growing, the amount of dung is also expanding, and that's an opportunity for not only the number of mushrooms to increase, but also different species can evolve as there's migration into different climates and different environments. So definitely the expansion of these herbivorous mammals should have impacted the distribution of the mushrooms, but also the underlying change in the climate that is sort of transforming the earth from maybe more forested to open grasslands.
Yeah, I think that's. I think one of the things that a lot of times just blows my mind when I'm thinking about evolutionary time. You're talking about psilocybe mushrooms or similar mushrooms to them, possibly having been on planet Earth, all the way back to Gondwana land. And I think one of the things that I'm cognizant of in terms of what's happening on a global level is climate change. And one of the things that we know about climate change is that regardless of how fast it's going right now, it is cyclical. And so the period of time that you're describing has multiple cycles of this glacial maximum and glacial retreat, cycles of, say, 20,000 or more, give or take, years. And during that time, even humans have been through multiple ice ages. Anatomically modern humans have been around for about 100,000, maybe 200,000 years. And so during all of that time, during the entire period of evolution of humans, mushrooms, in particular psilocybin-containing mushrooms, had been around for millions of years at that point. And so they're producing neurochemicals that we have found to be fascinating, valuable. We've considered them sacraments throughout our different cultures. And yet these very same neurochemicals have predated the evolution of anatomically modern humans, and possibly even, or very likely, the differentiation of human-like genuses, all the way back to even the evolution of larger mammals. So these neurochemicals are being produced by these mushrooms, and you say to interact with their competitions that they've got with insects and nematodes and the like. So I think that's fascinating, but I think one of the things that is hard to conceptualize, and I want to unpack with you a little bit, is that serotonin is this neurochemical that is most analogous to psilocybin. And I think a lot of people understand that serotonin is the target for a number of psychiatric drugs like serotonin reuptake inhibitors like Zoloft. So you're saying insects have serotonin?
Absolutely. And actually, before I looked into this sort of field, I didn't realize how widespread serotonin actually is in all of living systems. In fact, bacteria were the first organisms that were making serotonin, but serotonin has a huge number of roles in plants, in insects, in mammals. I like to think of it as sort of, if you think of the way that chemicals interact, there's an analogy of a lock and a key mechanism where a chemical will fit perfectly into its receptor and a response will happen. Well, serotonin seems like it's just a great key. It really fits into a lot of different locks and is used in a lot of different ways. Plants use it in not only defending against insect herbivory, but they also use it as internal signaling. It's involved in how they grow and how much they grow. It's very similar to plant hormones that determine how high and how fast the plant is growing. So it's in plants. It's in amoebas. Amoebas use it as a signal to grow in a particular direction or another, and it's also in animals. And animals is where it really takes off, in my opinion. So I was giving the analogy of the skeleton key fitting into a lot of different locks. That's what serotonin is. Well, in animals, they seem to have specialized a lot of different receptors that respond to serotonin. And what's interesting is that they sort of make specific keys. So, for instance, psilocybin really only interacts with one particular kind of serotonin receptor and leaves the other ones alone. But if you think about how those receptors are spread out throughout your body, it's almost like a fractal landscape where you have maybe 25 different serotonin receptor types, and they're painted differently throughout your body. Some of them will be lining your digestive system, and there'll be 3 or 4 kinds that are predominant in your digestive system, and they're responding to serotonin signals, and you're almost running a software program in there of how to move the food through your digestive system. But then you have a lot of them that are specialized to concentrating in different parts of your brain. So some of these receptors are potentially involved in different types of cognition. And what's interesting, a molecule like psilocybin targets just one particular, or maybe 2 particular serotonin receptors. So why? So if you look at insects, for instance, if you look at the fruit fly, Drosophila, which is the insect that we've studied for a long time because it lives in our garbage, they have that same receptor that's targeted by psilocybin, and where they keep it is in their subesophageal ganglion, which is a bundle of nerves that sort of takes in all the stimuli from their sensory organs and acts as a relay to determine what their responses are going to be to these different stimuli from their antennae and their olfactory senses. And it actually targets then that particular part of the nervous system of the fly. So then the effects that you observe when you directly interact with those receptors are decreased feeding. That particular receptor is also involved in the formation of memories for the insects. Their ability to identify which flavors they will be responding positively or negatively towards in the future. So it's really interesting that this compound has evolved specifically targeting one part of the insect's nervous system.
Yeah, it's fascinating, and as you were talking about Drosophila, which is it's something we've studied. It's a model species for those of you who aren't biology nerds. So we've done a lot of genetic studies, developmental studies on this particular organism. But I couldn't help but thinking of the old. What is it? Is a philosophical question about consciousness, which is, what is it like to be a bat? And, it must be like something to be a bat, and it's obviously different than what it's like to be a human. And so in this case, I'm thinking what it must be like to be Drosophila, the fruit fly, specifically under the influence of psilocybin. Is it anything like our own perceptual distortions? I mean, it certainly couldn't be all that much like it, given that we definitely have perceptions that we would imagine Drosophila not having. I think if you put yourself into the position of a Drosophila perception alone, we have that compound eye to mess with, which seems pretty psychedelic for default. So anyhow, the world may never know, but I think it's definitely an interesting thing to talk about, and you're providing us with little seeds for the imagination by talking about how psilocybin may be particularly targeting the feeding habits or the behavior of insects that you would expect it to interact with long before humans ever showed up on the scene. I think that I think one of the things that is not exactly something that you learn in your elementary school natural science or high school biology class is the idea that animals seek altered states of consciousness. It's a natural thing. Bees will seek out fermented grapes. We have video footage of foxes, for instance, taking huge bites out of Amanita Muscaria, and of course that same mushroom has a long history with reindeer. It seems, as I think Dr. Andrew Weil said, that seeking in intoxication is a natural human drive. You see it with children spinning around on the playground to get dizzy, and so I think it's interesting, that this drive seems to have been exploited not just by plants but also fungi, and we can see that it's a co-evolutionary relationship where there's a bait or a repellent compound that's being put out into the environment in these relationships that occur not just in space, but as both of us are evolutionary biologists, so this is occurring in populations over multiple generations in changing environments, and that the introduction of certain chemicals rearranges these relationships. Would you say that that's accurate?
Yeah, I think that's a really great place to let your mind wander in. There's the concept of evolvability is ways that you can adapt to new environments. And there's mechanisms underlying your ability to adapt to new environments. And we usually think of that as in terms of how our genes can adapt, because we think in a very neo-Darwinian framework where, it's particular genetic variants that are selected for. So if we're putting that in the context of psychedelics, it's really interesting to think that maybe the desire to seek out these altered states of consciousness is an evolvability mechanism that's happening at the level of culture or the level of outside of the body knowledge kind of thing. So if you are something that seeks out new ways of perceiving the world, you may come upon a new pattern that you can recognize. You may come upon a new idea that ends up seeding a way that your particular group of hominids survives or exploits its environment just a little bit better. And in that sense, it doesn't matter what the animal is that has this desire, this appreciation of intoxication. They could potentially alter their culture for their benefit by consuming them.
Yeah, so I'm taking in what you're saying, and I'm trying to, because I know your perspective, at least I have an idea of how you think about these things. And I think that, while we're talking about hominids here, we can easily talk about a number of, animals or arthropods that exist in populations. And when we're talking about, say, Hymenoptera, the broad kingdom, I think, or class of insects that includes bees and all the social insects, including termites, there are complex interactions between individuals within those populations. And so, these compounds, they have social effects. I think I'm fascinated with a lot of the bee and ant literature out there, and I know that if ants come back to the hive too drunk, they're punished. They get bit and punished, yelled at by their bees. So I think that there's complex interactions going on, but this altered state of consciousness doing something to the subjective perception of organisms that could be human, could be ants or termites, could be birds, but I think all of this does start to make me think a little bit about another thinker in the psychedelic mushroom world that most people listening to this podcast are probably familiar with. Terence McKenna, who spent a lot of time, shall we say, ruminating on these topics. And I think one of the things that he came up with that a lot of people have played with, and I think one of the great things about Terence was he never really shied away at speculating, wild ideas. He was a good generator of novel ways of looking at problems, and one of these is consciousness, but another one in anthropology is language. We know a little bit about language. We know that it's one of the ways that humans distinguish ourselves. We use to use a Terenceism, we use small mouth noises to vibrate the air, to vibrate an eardrum to transmit information, and that we have specialized vocal cords that allow us to do that. Our tongues can make sounds that even our closest evolutionary relations are unable to do. So in many ways, language is something that makes humans pretty unique. It could be argued that there are other languages being used, chemical signals, for instance, but it's something that we have spent a good deal of time trying to understand about ourselves. I think you go back to, for instance, biblical sources, and the Tower of Babel was a fable that was put forth to explain the really dizzying diversity of human use of language. And so Terence put together something called the stoned ape theory to postulate a way that language may have evolved. And his thesis essentially was that psilocybin mushrooms were a part of the diets of hominids out on the savannah. Apparently, there's maybe one or two levels to his argument. So first comes the grasslands and the following of ruminants in hunting, and he talks about some things about visual acuity that probably haven't withstood the test of time. But he essentially said, that the following of ruminants for hunting purposes by early hominids was something that would have brought them into contact with their dung while tracking them. When you're hunting, you're hungry, so you may have been eating insects or perhaps even mushrooms that were growing up in the dung behind them. And he then says, that culture then flows from this interaction, and goes one further to say that there was some kind of, and I, as an evolutionary biologist, I can say that some of his mechanisms are more than a little murky when he talks about how this all happened, but that some sort of co-evolutionary relationship occurred that may have resulted in human language. So that's a long introduction, but I just wanted to make sure that we're all somewhat on the same page here. Do you have any thoughts about this in the context of neurochemical ecology and co-evolutionary relationships?
I do. I don't know that it's exactly in the context of neurochemical ecology. It may be more in the domain of culture. I think the big questions, like why did the human brain grow to 3 times its size in 1 million years, I think these are amazing things. And any sort of way that we can put together the pieces, especially with something as intriguing as psychedelic compounds, gives us sort of a dopamine rush to be able to, potentially solve this problem. I think that I like the idea of psilocybin mushrooms as a catalyst, but I have to come back to really asking what are the drivers of this sort of evolution. So what kind of a role could the mushrooms have played in our development? I'm imagining it as much more subtle. So probably the major driver was the climate change. And when people come down from the trees, and I'm just going to use the scenario that Terence McKenna laid out here, the hominids came down from the trees and now they're standing upright. Well, as an evolutionary biologist, I like to think in terms of opportunities, limitations, constraints, and one of the constraints perhaps to an organism to have its brain grow in size is the fact that it's the same area where the brain would be growing is part of the animal's ability to get around. If you grow this large cranium on a quadruped or on a partial quadruped, it's going to get in the way of your mobility, and there could be problems with that. But when a hominid comes down from the trees and is in these tall grasses looking around and adapts to being in that environment by having forward-looking eyes, which are really very much towards seeking and finding new places where there's new food sources. Well, if you're standing upright, then there's no longer any constraint for the brain to expand behind you. And if that expansion is happening, you're also seeing where the largest part of that expansion is happening is in the visual processing parts of the brain. So if you can imagine this hominid that's looking out over tens or hundreds of miles of grasslands, scanning a huge area, lots of input coming in, trying to pick out tiny little signals of, maybe a gazelle or maybe some other source of food, there's a lot of demand for visual processing driven by food, right? If you can process that entire landscape and identify which direction to go that you're going to find the food, that is a major driver for selecting for the ape with the bigger brain.
Yeah, that
Where
makes
did the
sense.
mushrooms come in, though? I mean, that's the question.
Yeah.
So I think it's probably, if they do play a role, I think it's much more subtle. So the ape comes along and eats a mushroom. It has a psychedelic vision. Perhaps it gets an idea that it wouldn't have had before. Perhaps it understands its environment a little bit differently from distorting or reconfiguring the way that it normally interprets its environment, and that becomes, maybe we'd call it a meme. Maybe there's this meme now that is actually useful. It's an idea that helps the ape maybe acquire more food or expand into more territory. Well, now you have a demand for evolution of the brain to better do whatever that is, to better manifest that idea. So language could be one of those things. So you could have had as a catalyst, as you say, but not necessarily as a driver. So the driver will always be ability to reproduce, ability to acquire the resources in order to reproduce.
Yeah. I think I'm right there with you with a lot of this. Climate change is what we would call a big force. It's rearranging relationships and defining how available certain kinds of resources are. It's changing whole ecologies. And again, if we're to think about this grassland scenario that seems to be at the heart of what most people have a consensus of, a part of humans' evolutionary history, the access to food, changes in locomotion. These are again big forces, and Terence tried to engage with some of these things by saying, well, it could have affected fitness by having large orgies or something, and I think that that may not be the biggest factor here. I think that there's been a lot of great books out there on the sexual ecology of humanity. Sex at Dawn comes to mind, that puts together a lot more coherent histories of the sexual ecology of humans and our close relatives. But I think one of the other things that you didn't mention was that, as far as I'm aware, humans have had fire for as long as we've been anatomical humans. And so that would mean that Arthropithecus, if I'm pronouncing that correct, some of the other Homo erectus, our distant, our close relatives who are no longer with us, would have had fire at some point along the way, and that there was somebody at Harvard, I believe, who said that cooked food, despite sorry raw foodists, but cooked food is more bioavailable. It's easier to digest. Our close relatives, chimpanzees and bonobos, spend a lot of their time eating because it takes that time to masticate, to chew, to process, and to liberate enough nutrients to survive and grow. But when we started using fire to cook food and meat, we were more efficiently liberating nutrients and had a lot more available nutrients to mess with, and that this I think was one of those things where if there's not as much of a limit on nutrients than other limitations on a trait like brain size. Many of us are aware that your brain is one of the biggest sinks for your caloric intake. And so I think these forces, go a lot more distance in terms of explaining some of the mysteries of human consciousness than mushrooms itself. But I think that we can probably thank Terence for really, through a kind of take no prisoners, any means possible kind of way, refocusing the conversation a little bit to say, hey, psilocybin is drastically altering our consciousness and probably was in the pharmacopoeia of ancient humans. What might that mean? And I think for that we can thank Terence. But I think we would have been remiss had we not talked a little bit about the stoned ape theory. So thanks for engaging a little bit.
I totally agree. I think it gets a lot of our creative ideas flowing, and it brings a lot of people into thinking about ecology and evolution. I'm happy that it's out there.
Yeah, awesome. So I wonder, we should probably talk a little bit more specifically about the work that you've done and the contributions that you've made more recently about our understanding of psilocybin and where it all fits from this neurochemical ecological perspective that you take. So before we really get into the specifics, I feel like we need to back up and talk a little bit of vocabulary here. So
Sure.
in the title of your paper, you reference something called horizontal gene transfer. And I wanted to ask for our listeners and for my own edification here, exactly what is horizontal gene transfer and how does it relate to psilocybin?
Okay, well, there's a lot there. So horizontal is the opposite of vertical, I guess. And vertical transfer of information means from parent to offspring. So you get DNA information to produce you from your parents, and it gets passed down from generation to generation. That's called vertical transfer of information. Well, horizontal is not happening from parent to offspring. Horizontal transfer of genes, horizontal transfer of genetic information is happening between species. Somehow, one species is copying genetic information from another species and potentially using it in the same way or a slightly different way than the original species was using it. So, for example, if you have two bacteria in a hospital when they're using antibiotics to cure, I don't know what they use antibiotics for exactly, but one bacteria has a gene that enables it to resist the antibiotic and the other one doesn't. Well, sometimes bacteria can slurp up some DNA from their environment or actually have it actively transferred from one bacteria to another. And once that genetic information is now in that other bacteria, it is resistant to the antibiotic. So the transfer of antibiotic resistance is the classic example of horizontal gene transfer.
Okay, so I mean that's fascinating on a lot of different levels. I think one thing I wanted to talk a little bit about is that, or at least just note, is that, hospitals are actually a breeding ground for antibiotic-resistant bacteria. I think the one that I'm thinking of in particular that's a problem is antibiotic-resistant staph, so Staphylococcus. And so that's an environment that is selecting for precisely the kind of transfer of information between, there's a bacteria that has figured out a solution to this problem, and there is a tremendous selection cost for not having figured out this problem. And so the solution is there in the genetics, which codes for some kind of enzymatic resistance, or some other way, and then it's passed from one population or one individual to another, and this can occur in bacteria. I think some of us can remember the plasmid is one of the ways that happens. But so you're saying that horizontal gene transfer is something that can happen in multicellular creatures like mushrooms. That's fascinating.
Yeah, we think it's quite a bit more rare, but we have a lot of evidence that it is happening. In fact, that's been a huge amount of my literature that I've put out into the world is demonstrating evidence that fungi are exchanging genes between different species. Some of these genetic exchanges seem to be quite important in the evolution of lineages. And this is one example in the paper that you're talking about having to do with psilocybin, where we think that one species of mushroom benefited by getting the ability to produce psilocybin from another species of fungus.
So I read your paper, and I I know that what you're talking about from your paper is that psilocybin came originally, or at least as far as we can tell, from the Psilocybe species groups, that genera, and that it was then passed to another genus of mushrooms, that some of these species have psilocybin and some of them do not. So Paneolis would be the genus, and it's funny, you can find Paneolis mushrooms. I found some out front of our building here. They're quite common in grass, and so they did not inherit this biosynthetic pathway to produce psilocybin from a parent relationship. They somehow received it from directly a totally different genera.
At some point back in their history, yes. So I'm sure the evolutionary story is really complicated, but the simplest version that we can come up with is this. So we think that the wood decay psilocybes probably were if not the original producers of psilocybin, maybe they were early producers of psilocybin. It's widely distributed amongst psilocybes that decay wood in fairly rotten environments. They like rotten wood, they like wood chips, they like buried wood, this sort of thing. So we think that maybe that particular environment where the plant matter is really decayed and it's really built up with a lot of the nastier plant chemicals that plants produce for defense, and it gets harder and harder for fungi to break them down, but it actually gets easier for things like insects to burrow in there and kind of chaw away at fungi, which are pretty nutritious. So what we've gleaned from reconstructing the evolution of genes, and I probably won't get deep into that because it gets a little complicated, what we've reconstructed is that that from those ancestors, when the environment shifted after the KT extinction, you had the emergence of grasslands back then too, and this gave rise to some of these mega mammalian herbivores, even within just a few million years.
Can I stop you really quick and can you just mention for the listeners who may be not aware what the KT extinction was?
Sure. At the boundary of the Cretaceous and the modern era, there was probably, I think the best idea is that there was a comet impact. It caused a lot of havoc on Earth, a lot of climate change, and the extinction of all the dinosaurs pretty much except for birds.
It hit like in the Gulf of Mexico, as far as people think, right?
Yes, but the continents were in very different positions at that time.
Right.
So what that exactly
Everything's
means,
always moving.
Exactly. So after that point, you no longer had these large reptiles and large birds. And what filled that space, and some people have argued that they had to be warm-blooded because of fungi that were able to feed on dinosaurs, on cold-blooded dinosaurs, but that's a totally different story. What filled that niche space, along with the expansion of grasslands, were these large mammalian herbivores, and they got like 10 metric tons within just 5 or 10 million years. You had these mega herbivores, no doubt putting out hundreds of pounds of herbivore dung on these open grasslands. So the evidence from the evolution of these mushroom species is that dung-loving species evolved from those older wood decay species because the environments of dung almost exactly the same. And we looked at the genes that are involved in decaying dung and decaying rotten wood, and they're almost exactly the same. You can't really tell them apart. So we think that from there, a lot of different fungi evolved to exploit that new niche of a lot of mammalian herbivore dung. Some of them were psilocybes, some of them were Paneolis, which you were talking about, having received this cassette of genes from Psilocybe. Well, the idea, we have evidence that the genes to make psilocybin were acquired by a dung-loving Paneolis from a dung-loving lineage of Psilocybe. So when you have horizontal transfer in dung, you can possibly suggest, just like when you have horizontal gene transfer under an antibiotic, that there's a really important selective pressure that's driving that.
Yeah, so that environment, it's complex. There's competition from insects. There's danger, and all of those dangers affect the ability of any given species, fungi in this particular scenario, to proliferate or to be excluded. And so, if I'm understanding you correctly, the ability to survive in that particular environment was made easier by psilocybin. And so you're talking earlier about what we know from Drosophila and the feeding behaviors. So the chewing that might have occurred, I think we'll never know what it's like to be an insect, but we can see, the idea that in this space that we're describing, you might have the option to eat dung or other things living in it, or mushrooms. And you might prefer mushrooms over poop. So there was this, inclination for insects to engage in mycophagy, and obviously that's bad for the mushroom. And so, when I was reading your paper, I was sort of brought back to some of the basics of my ecological training, and I thought about how ingenious this was as a solution for this long-standing relationship. I think most people know that dung attracts flies, and that relationship has probably been stable for a long time. But I was thinking about the red queen hypothesis and about how there are evolutionary arms races that occur when your opponent is chewing on you or murdering you, and you choose a weapon that murders them. Well, that then instantly becomes a selective force on their evolutionary story. And so psilocybin is one of the safest recreational drugs that we know of, according to a number of recent global drug surveys. They're very low LD50. It doesn't kill even in massive doses. And so I think it's. Can you comment a little bit about that over time?
Why is it a really safe drug for human beings, but not necessarily? And why does it still make sense that it was an evolutionary advantage to the fungi fighting with insects? Is that sort of the question?
Well, I guess the impression that I got was like, this is ingenious because psilocybin is probably effective at confusing insects that would like to eat them, but because it's probably, as far as I'm aware, it's not fatal to these insects, right?
Well, fatal is quite relative, and especially when you're talking about things that live for a few days or a few weeks, and fitness is driving everything. And when I say fitness, I mean your ability to put eggs that are fertile into the next generation. That's how it works. So if a fly is slowed down ever so slightly, then it's going to not make as many eggs as its neighbor who didn't chew on the psilocybin, and it may favor some sort of behavior in the insect to recognize psilocybin and go turn to the other fungus or the bacteria that don't make it. So it could just give a little bit of opportunity to the fungus to grow more and create larger mushrooms, to have more spores, to find more cow patties. And at the same time, it encourages the fly to eat something else.
Okay, so it may be that some insects out there have had long association with psilocybin, and they're perhaps even immune because it was a selective force in their
not
past.
Only immune. I mean, there are other toxins that mushrooms produce that you can't imagine anything overcoming them. Amatoxins, for instance, made by Amanita. It interrupts one of the central parts of cellular metabolism. And there's a certain species of fly that has a mutation in a gene that enables them to eat that mushroom tissue, and they actually find that particular mushroom, and they lay their eggs there, and the eggs go through their full development on that mushroom. And they have absolutely no competition.
Yeah, it's a safe place.
Because,
So am I correct that they call that the destroying angel, that particular Amanita?
Yeah, it's a destroying angel.
Yeah. Careful out there, mushroom hunters. A lot of times people mistake it for matsutake. Anyhow, thanks for unpacking some of that, because I think that, I don't think that we often put ourselves into the position of what it's like to be a fly running around in a cow patty, and yet, the psilocybin molecule, for those of us who have experienced it, is something that it presents more questions than answers from an experiential level. And so one of the things that I found so fascinating about your work is that it really sits with it and tries to come up with a well-reasoned, evidence-supported hypothesis for the ecology of and evolution of this particular this trait that causes such profound changes in our perception, and then provides a context-specific way of talking about it. I wanted to talk a little bit more about your methods. Not everybody really understands. I certainly don't understand a lot about genome sequencing and identifying gene clusters. Can you talk a little bit, without going super technical, about how it is that you came to identify the genes that were involved in psilocybin, and then how you were able to show that likely this host of genes had been passed across into other genera?
Answering the discovery question is a lot easier than answering the evolutionary question. So the discovery is a very simple story. It was an idea that just popped into my head. I was talking to a friend of mine at a mushroom conference at Yale, 6-7 years ago. And when the genomes were coming out, the ability to sequence genomes was becoming more and more easy. We thought, hey, nobody has really figured out how psilocybin is made. It seems like something that we should have known a long time ago. And we don't, because for the past 30-some years, it's sort of been poo-pooed science. And, people have tainted their careers and gotten into... It just hasn't been a favorable environment for doing that research, I guess I would say. So that changed sometime, in the mid-2000s, as psilocybin was starting to take more potential therapeutic routes.
Yeah, the Hopkins work really started to take off around that.
Yeah, University College of London. So it sort of opened up a space where we could start to think about this again. And the obvious question is the genetics underlying this and the ecology underlying this. So I sort of diverted from the question there, but all we did is we sequenced 3 very different species of mushrooms that make psilocybin. And then for each of them, we found another mushroom genome that was very similar, but didn't make psilocybin. And then we just compared them, and we found the genes that were only in the psilocybin-producing mushrooms. We ended up with about 30 genes. that was common to those very different mushrooms. So how do you find out of those 30, you still got, it's not a haystack anymore, but so then it was actually a very quick step to finding 5 of those genes were in a straight line on one particular chunk of the genome. And we looked at the functions of those genes, and they were everything that you needed to make psilocybe. So we identified it. But then we had to go ahead and prove that it was doing what we said it was. So we had to do some testing of the enzymes to see if they would make the products that we thought. Because my lab doesn't have permission to produce psilocybin, we were limited in which enzymes we were allowed to produce and test. So we were only able to look at a couple of them. But at the same time that we were doing this work, unbeknownst to us until we were about ready to publish, Dirk Hofmeister's group in Germany was busy. They had also found the gene cluster, and they were busy characterizing all the genes while we were working on the evolution. And we tried to coordinate our publications, but his peer review was a lot faster. And anyway, so that's how we found the gene cluster and evolution.
I think there's a lot there that I want to touch on, and we'll get back to some of the other questions if we remember what we're doing. First, I just want to note that it's always fascinating. It's something that happens throughout the history of science. Even going back to Darwin and Wallace. You had I guess there are just these moments where evolutionarily speaking, we would call it convergent evolution. You have 2 different groups at the same time working on the same problem and coming to the same conclusions. That kind of
different methods.
Very different methods. Yeah, that I haven't read his paper, but I'll have to take a look at it. It's just fascinating the way that technology sometimes creates the conditions where things happen at the same time in a synchronistic sort of way. I mean, it sounds like you're processing, a tremendous amount of data, but there are new tools for driving down the burden for sequencing and comparing whole genomes here, and narrowing down to tiny little fragments of that alphabet that has 4 letters.
Yeah, so it's a lot of information that's processed, but it's really doing very simple tasks over and over again, which is enabled by a computer. So all we really are doing is taking 2 little pieces of DNA and comparing them. Are they similar? Yes. Okay, but you got to do that with millions and millions and millions of pieces of DNA. Well, the computer does all that when we write a program that ends up sorting all the DNA into a couple of little boxes. And this box is the genes that are only in psilocybin-producing mushrooms. And this box is all the genes that are only in dung fungi, for instance. But the next step is trying to understand, okay, well, they all have those genes. How did they get them? And that's a different kind of analysis. That's reconstructing the evolution of the genes. So we're not only comparing one gene to another gene, we're building an evolutionary story of how the genes that we see now came to be, how they're all related to each other. So what we found was that all 5 of those genes that I was telling you about, all 5 of them had the same evolutionary story. And that story was that 2 dung fungi, they were very distantly related, 2 dung fungi had the most similar genes, whereas 2 psilocybe mushrooms had somewhat different genes. So the dung fungi were much more similar, even though they were distantly related. That is a classic signature of horizontal transfer having happened perhaps in that environment, maybe because of some selective pressure in that environment like we were talking about before. And that's where we led into speculations on it was insects. They were probably driving that because insects are a huge selective pressure in not only that environment, in the dung environment, but in the wood chip environment, in the in the buried wood environment.
Fascinating. You validated the biosynthesis of psilocybin. Is that something that you did by cloning those genes, putting them into E. coli, and that was why had you taken it all the way, you'd be manufacturing psilocybin in a petri dish? Is that correct?
Pretty much, that's the story. Yeah, and actually, so we were initially thinking that the biosynthetic pathway was what they thought back from 1968, the Arguello paper, I believe it was, where the synthesis was, how does this work? The decarboxylation, and then the next step would have been N-methyl transfer, and putting the phosphorylation of psilocin to make psilocybin as the last step. That is the canonical pathway. For a very good reason, we know now, that's the way they thought that psilocybin was made. So we were going by that. So we were thinking we would take the first two steps, which would make non-controlled substances. So we cloned, like you explained, we cloned the decarboxylase step, the first step, and we expressed it in bacteria, and we fed it some tryptophan, and it made tryptamine.
So I wanted to point out or draw a parallel here. So you used a method, a transgenic technology, to validate your hypothesis. And correct me if I'm wrong here, but transgenic technology sounds like human-mediated horizontal gene transfer. So what I think is so fascinating, and one of the reasons why I like to study and think about nature, is that we're talking about a trait that was passed horizontally, transgenically, without the intervention of any university laboratory. There's tremendous ingenuity in the natural world, and am I correct that we don't exactly know how this transfer happened? I think there's a couple of methods that can be mediated by bacteria or viral transmission, but there seems to be. Is there some particular way that mushrooms are able to do this that we understand, or
there's not a smoking gun in fungi? There's lots of suggestive bits of data. My personal bias is that when fungi are under stress, they slurp up DNA from the environment, and sometimes it works and sometimes it doesn't. They might get something from the environment. Well, if they picked up something from that environment, chances are something else is using it for some purpose in that environment. So you're actually acquiring an ability that's useful right there, right then. So the chances are greater there that in that situation, that you're picking up something that's going to be useful. But as far as how it happens, no smoking gun in fungi. People have, we've seen evidence of transposable elements. These are bits of DNA that jump around within the genome. They sort of hang out on the edges of genes that have evidence of having been transferred. There's evidence of DNA forming circles, which stabilizes the DNA so that it might last a little bit longer when it's got these transposable elements. It's more likely to survive a transfer event. But nobody's really demonstrated viruses transferring in fungi the way you might see in bacteria. Nobody's demonstrated a specific mechanism that's evolved to specifically transfer. But at the same time, it's really easy to artificially induce fungi to take up DNA.
Interesting. So they just they slurp up DNA. They're somehow directly interacting with this raw data that's just laying around in the environment. I think that's fascinating. Just from my own subjective experiences with one of the traits that psilocybe mushrooms are producing. psilocybin can blow up your senses, and it can actually feel like you're interacting with an environment in a much more direct way. There's probably no logical link to that, but I'm doing a little free association here. So I'm cognizant of that we should probably talk a little bit about the other fascinating project that you have been a collaborator on, if I'm correct in that. And so can you tell us a little bit about this project involving cicada parasitism that you've been working on?
This is a really fascinating story that I just lucked into being involved in. So there's these cicada pathogens that infect the cicadas. I'm not the entomologist here, but this is work that is organized by my colleague Matt Casson at West Virginia University. These cicadas are infected by a totally different group of fungi that has absolutely nothing to do with mushrooms. He diverged from mushrooms maybe 1 billion years ago, something like this. They infect the cicadas sometime before they emerge from their long time underground, and the cicada's entire rear end becomes transformed into the spore-producing structure. So they're flying around and they're dripping these white spores. So in the Casson lab, they call them the flying salt shakers of death, I believe. But what's interesting is there's a couple of species that they've looked at of this fungus pathogen of cicada, a couple different species of the fungi, and one of the fungi produces psilocybin in this plug.
Fascinating.
And another species doesn't produce psilocybin, but it produces cathinone, which is an amphetamine.
Also known as bath salts for all of you out there.
Oh, is that the common name for it? Okay. So yeah, you've got these related species of insect pathogens that are somehow, we don't know exactly what they're doing to the cicadas, but we don't know what the chemicals are doing to the cicadas, but it's an amphetamine and it's a hallucinogen that are being used, and they are modifying the behavior of these insects. For instance, the male cicadas change their behavior to make a clicking that resembles that of a female cicada. So you'll get mating between the male and the infected male, and this is actually a mechanism by which the pathogen is transferred from one cicada to another. So they're modifying the behavior, and it's increasing the dispersal of this particular fungus.
So just to be clear here, we're talking about homosexual interactions between insects where there's effectively a mushroom replacing the rear end of one of these insects, and this is passing on the spores of the fungi.
Yeah, absolutely. So this is behavior modification of the insect by the fungus, and what Richard Dawkins would call this is the extended phenotype of the fungus. Essentially, part of the fungus's phenotype, its manifestation of life, is the manipulation of this insect. It doesn't kill the insect, which is kind of interesting. You'd think, why not just kill the insect and feed on it? Well, the insect is specifically kept alive, and these rear ends that are nothing but spores, they fall off, and the insect doesn't necessarily die. It's the benefit to the fungus is that these mating attempts happen to spread the spores. More spores spread, more fitness, and that's how evolution works.
Yeah, it's certainly still a bad deal for the insect because, in evolutionary currency, if you're not leaving offspring, you're not going to be around for very long. So there's definitely a cost in that environment. And so just to clarify, so the cathinines, which I think are also produced in a stimulant that's used in Middle Eastern cultures, very much like coca leaves, called cat. Now this is in a different but related species. So it's using cathinines to modify or presumably to modify the behavior of these cicadas, whereas another one is producing psilocybin. So do you think, have you explored in this work very much, like, do you think that the psilocybin may have come also through a similar process of horizontal gene transfer, given that, cicadas spend a lot of time underground where they might come into contact with, some of these wood rotting and dung rotting species?
Well, this is actually how our lab got involved, because we're the ones that look into the genomes. And the problem with answering this question in that particular system is that you can't grow these fungi in a petri dish. You can only find them when the cicadas emerge, and you can't get them pure. So when you sequence the genome, you try to find the genes, you also get a bunch of insect genome in there. So it's really hard to really interpret what genes the fungi actually have. So we sequence what's called a metagenome. It's the insect plus the bacteria that are there, plus this particular fungus. And although we didn't do that in my lab, another collaborator in the group did that. But we sort of filtered out the genes that we thought were the fungus, and we looked for evidence that it was the same genes that were in magic mushrooms. We found no evidence that it was the same genes that were in magic mushrooms.
Fascinating.
They must be, unless we didn't get all the fungal genes, there must be an independent evolution of making psilocybin, which kind of argues that, wow, this is a really special molecule. If in 2 billion-year diverged lineages of fungi, it's evolved this bizarre modification of a tryptophan amino acid has evolved twice. That's really pretty interesting. And in both situations, well, one interaction with insects is speculated. The other interaction with insects is direct. It's involved in pathogenesis of insects. So why psilocybin?
Fascinating. For those of you out there that have been watching nature documentaries or are familiar with the work of Paul Stamets, you may be familiar with these other species out there called cordyceps. That whole genera is, a parasite on everything from tarantulas to caterpillars to ants, and they also modify the behavior of these animals, these arthropods, and so. This is some kind of neurotransmitter mimicry that we're talking about here, and it obviously has some kind of utility, and we see it in both plants and animals. I mean, there are certain species of a number of species of mimosas and acacias who are producing large quantities of these neurochemical mimics, in particular dimethyltryptamine, which looks very similar to serotonin, and it can be up to 2%, 3% of the weight of these roots of that tissue, and from a perspective of investment, that's quite a lot of investment in a particular molecule. So we've talked a little bit, I think. So to talk about this, I want to, back up a little bit. There was a really great reference in your work to some relationships with termites, where these fungi are physically mimicking the eggs of termites, and potentially also exuding some kind of compound to interact with and disrupt the behavior of termites. Termites are social insects. They have complex societies. They can even, I think, not just termites, but other social insects have complex societies. And they've been said to even have invented forms of agriculture and animal husbandry, in particular with their relationships with fungi. They cultivate them. Some leafcutter ants will feed and house and cultivate fungi, and then ant relationships with aphids. That would be the animal husbandry thing. So on this level of nature, there's really vast social organization. And then when we think about the neurotransmitters that are just sort of floating around in the environment, whether they're psilocybin or dimethyltryptamine, or even some of these anticholine compounds like muscimol and other secondary compounds that are being put out into the environment. This is a complex environment. It's interacting with the psyches, the subjective experience of these insects. But let's bring it back in, Brian. The neurotransmitter mimicry. It's a thing. Like what?
And there's so many neurotransmitters that can be targeted. So there's not only serotonin, there's dopamine, there's GABA, there's all these different ways. And in the zombie ants that you were talking about a minute ago, they use the GABA pathway essentially to control how much inhibition, presumably, the ants have. It modifies the behavior for these ants to climb up to a high point and before they bite down and die. And we don't know how these termite egg mimicking fungi, how that exactly works. It's a very young study, but we were clued into it being possibly involved with psilocybin based on our paper. What we found is that they call them termite balls. These termite balls fungus, they're the only other fungi that we found in a database of hundreds and hundreds of genomes that have every single gene required for making psilocybin. So we thought,
fascinating.
What's going on here? We don't know if it's making psilocybin, although we're actively investigating that. And it is known that these they make these little fungal balls, and they have a surface coating on them that's actually an enzyme that's involved in breaking down wood. But termites use that enzyme as a pheromone. It's given off by their eggs as well, and it causes them to take care of their own eggs, and that enzyme tricks the fungi into taking equal care and piling them up and incubating these fake eggs just like they're their own. But we have to know, personally, I have to know, why do they have all the genes for making tryptamines? So that's an active area of investigation.
Wow, we'll be definitely watching that one, and maybe we can have you back on once you figured it out. I think one of the things that fascinates me is with all of these neurochemical mimics out here. They're producing them, and it seems that our understanding of them is that they're signaling molecules, or that they're mediating the behavior of other species. But do we know whether or not plants or fungi are using any of these neurochemicals like in-house? Are they using them for cognition? Are they using them for signaling internal, or are they only outwardly directed molecules?
Well, I think they're both. I mean, there's evidence that serotonin is used by plants and amoebas and lots of different organisms. But as far as the mimics, I think that's where you're probably getting into a territory where these are specifically being selected for because they're having some sort of ecological role. Why would you interfere with those pathways?
I had a big association there where, something that is big in the studies of artificial intelligence is the Turing test. Whether or not a computer can convince you that it is in fact human with its own thought processes and all that. And when you're talking about these termites, we might imagine that these termites have become convinced that they're dealing with another termite, or at least a baby termite. And I mean, we see examples of this in mimics throughout the literature, but it's just
termite Turing test.
Termite Turing test, yeah, the TTT. I mean, I think that these kinds of discussions, they make me go, what does all this mean? Like, where does this literature take us, and what can we learn from these big questions?
Well, it's complicated. Seriously, we live in an extremely complicated environment, and there's just so many ways of doing things and so many ways of figuring out your environment, of being within the environment, of communicating, of interrupting something else's communications. And is there one central meaning to this, one central meaning to life that we're going to find? I don't know. I mean, we certainly see patterns emerge. We certainly see convergence on very similar chemical structures of the meaning of life, certain types of chemical structures. Probably not. But there's probably some thermodynamic principles underlying those specific chemical structures emerging. And why are some things signals and other things are structures? And but I think it's just that it's really fascinating and beautiful and complex, and we'll have lots of interesting puzzles to solve for a real long time studying these things.
Yeah, well, thank you for all of your insight and sharing your research with us. I was really fascinated by your work and wanted to spend a little bit of time unpacking it with you. So thanks again for coming and making some time to chat with us.
Oh, thank you very much. I've enjoyed it very much.
Awesome. Great.
Thank you for listening to this episode of Psychedelics Today with Dr. Brian Pace and Dr. Jason Slott. It was a great episode and hope you learned a lot. If you want to support the show, leave us a review on Facebook or iTunes. Please, please, please. We'd love it. And if you want to get educated, check out Navigating Psychedelics over at psychedelicstoday.teachable.com. We'd love to help you get educated and reduce harm. Education is harm reduction, and you can reduce the impact of the harm of drugs just by learning. You could even call it risk reduction, since a lot of these psychedelic compounds have very low risk or harm profiles. It's not really too much harm there. So check it out. Navigating Psychedelics over at psychedelicstoday.teachable.com. Hours of lecture and 10 or 12 master classes. Hope you enjoy the show. Take care, everybody. See you on the next episode. This is Joe Moore signing off from Breckenridge, Colorado.